Log on, tune in, find out. Another good idea from Cambridge.
Same problem.
Why do you assume so?
So even better.
I don't want to promise anything.
So next talk is by Maciej Patan and Basia Bogacka.
Yeah, well done.
Dose selection incorporating PK/PD information in early phase trials, please. And again, please make use of the nice format and chip in, ask, comment as we go along. Yeah?
No stakes. No stake.
Sorry? No, she doesn't have a stick, so you can ask. So people in the back row can ask.
Well, thank you very much, first of all, for inviting me here and for the introduction. As you see, this is the title. So what I will talk about, of course, this is my joint work with Maciej, so that's obvious from what you've seen the first slide. First two points, I think they are unnecessary here, so I will go through very quickly and then go to modeling. So as in the title we say, it's in PK, we add PK/PD information into dose selection. And all of it is model-based. So I need to say something about models. And there is first in this modeling section, dose-response model, but actually everything is dose-response in a way. But that will be about modeling the probabilities of success or failure. And then PK, then PD. PK is for pharmacokinetics, PD for pharmacodynamics, and then the joint model, PK/PD. And how do we use it in the design? The design is adaptive. So I will say what happens in the case step of such a design in our case. And then this is an optimum adaptive design. So of course we need some optimality criteria. Oh, please sit down here.
Possibly move like 1 or 2 chairs in. Somebody is sitting on the floor down there, right? Sorry, thank you.
Okay, so that will be a little bit about the optimality criteria, and this will be constrained by some requirements for efficiency of estimation and for toxicity probabilities. And then I will show you what happens with this adaptive design. So clinical trials, we have all these phases very quickly. Why do I show this slide? Not because I want to teach you about these phases, but because I want to tell you where we are. Around here, probably. I don't know, because phase 1, I'm an academic, so my experience in real life is very limited. I'm sorry about that. Well, that's true. In this, of course, in respect to clinical trials, you understand. Otherwise, that's a different story. So, in literature, what I find is that phase 1 is first in human, usually healthy volunteers, and very small. And what the main interest is tolerability, pharmacokinetics as primary purposes of doing these trials. Then in phase 2, I found recently some information that actually there may be phase 2A and phase 2B. I don't see much difference myself between these two, but phase 2A says something where it's already in patients and pharmacodynamics is already of interest, that some biological activity and also clinical efficacy as well. So there may be more information added, but still we want to work on dose selection. Then some definite dose range finding in the second part of it, and then phase 3, very large confirmatory studies. So I think that the work we try to do here would probably fit best around phase 2A. Okay, so this is early. So we want to establish PK and PD parameters and to find safe and efficacious dose to recommend for further studies. And it doesn't have to be just one dose level, maybe more, but we will here focus on one. And what would be observed altogether? That would be concentration of the drug candidate in plasma. That would be a PK. And that's a continuous kind of on a continuous range. Then the response, well, as I said, everything is a response to the drug candidate, but that's what often in literature is taken as response. Is there a toxic effect or not toxic effect, and then probabilities of these events are modeled. And then some markers or surrogate endpoints. And these markers or these surrogate endpoints or pharmacodynamic ones may change in this setup over time. So that, for example, would be like you get a drug into a vein and you measure pulse. And of course, depending on the plasma concentration, this pulse will change over time. So that kind of PD endpoints we take into account. Okay, so as I said, these are adaptive designs. And what are the main features? Well, just very quickly, the information is gathered sequentially. After each step of the trial, so we have several steps, the statistical model is updated. So first of all, as I say, we assume we have a model. And it is updated, which means that this model's parameters are estimated. Okay, each step. And then next step of the trial depends on the result, which we have up to this very moment. So all of them from time one.
Looks awfully Bayesian to me.
Yeah, well, I said it may. And then the trial is stopped according to some pre-specified stopping rules. And I don't say what they are here. I will just show what they could be on for the example. So modeling now. First, let's look at the model I'm taking into account. But again, that's not the only possibility. There are lots of possibilities, and we have many people here who work with slightly different models. I don't think that this would change very much with these different models. The idea would be roughly the same, but I took this as to follow as an example. So we have three responses, or three categories, Y0, Y1, and Y2. And Y0 is that there is no efficacy, no severe toxicity, sort of neutral response. Then success. So there is efficacy, but no severe toxicity. That's what we are aiming for. And then there is severe toxicity. Whatever, is there efficacy or not? I don't want that. That's a failure. So this is the approach. Then each of these has assigned probabilities, psi0, psi1, and psi2. And this will depend on the dose, x. X is a dose, and this will depend on some parameters. Theta1 is a vector of parameters. And then, again, here it could be different modeling of how these probabilities depend on the dose. This is not the only way. It's one of many possibilities. That's what we've taken into account in this work. So these are the relations of the probabilities and the dose. So we have four parameters, alpha 1, alpha 2, beta 1, and beta 2. This is my theta 1, and this is how the dose is related to the probability. So when you solve these equations, you get these probabilities, and these are these three nonlinear models in parameters. And depending on the values of the parameters, you may get various shapes of these functions. And here are some examples. So what is here? This is psi1. Psi1 is for what we really want. So this is probability of efficacy, no toxicity. Probability of toxicity is this line, here, here, and here. It's always increasing. But the probability of success may have various shapes. Here is non-monotonic, and here is decreasing. That's not ridiculous. That may happen. So depending on the values of parameters, we may have various scenarios. In further examples, we will look at this. But the same calculations can be done for any of these scenarios. What we want to estimate the dose to be recommended for further studies. And as I said already, this is probably okay for this approach for patients, when we have patients, because we are interested in efficacy and PD endpoints. So healthy volunteers, we might just simply not observe it. And the numbers which we'll need for these trials are not that small either. And then the questions we set here is what strategy to take to apply these efficacious doses. Efficacious doses. That's an important point here, not any doses. We want efficacious doses as often as possible. Of course, we will have to apply some which are non-efficacious or which are toxic, but we would like to have as many efficacious as possible. To get this, and based on that, we want to recommend something for next studies. And also how to incorporate this pharmacokinetic and pharmacodynamic information.
And when you say efficacious, you mean just to have a marginal efficacy irrespective
Yes.
of safety?
No. Efficacious for this is no toxicity.
Efficacious
Yeah.
and no toxicity.
That's right.
What you
The
That's
two
right.
events are exclusive. They can't both occur in the model.
Yes. Okay, so PK model. Again, here you may have various situations. These are usually some compartmental models. This is the simplest, or probably not the simplest, but a simple example of such a pharmacokinetic model, which is a mechanistic model. Here, as a set of differential equations, in brackets, there's concentration. So here we assume that concentration at time 0 is 0 for these two. A would be drug and B would be metabolite of the drug over time.
And lambdas are
Lambdas
general
are
powers?
Lambdas are known in our case. In general, they are unknown. Usually what you find in literature is that lambdas are 1. What we've taken here for this example is that lambda 1 is 1 and lambda 2 is 2, which makes it a bit more complicated, but also more interesting, in fact, from mathematical point of view. Is it practical? That I don't know, but I know I've talked to people on some other occasions if lambda equal 2 is sensible, and I heard that it is. So this is not, again, uncommon, completely uncommon perhaps.
I'm sorry about the initial condition. For me, A at 0 should be equal to the dose given, to the input you give into the system.
Well, yes. In fact, we have a dose infusion rate incorporated in it, which is here. Okay. So we wanted to add the dependence of A and B on dose, and now solving this set of equations we get. Here's a function of time for a given dose, and we've chosen this function to be just proportional to the dose. C is some proportionality value, and it is a drug infusion rate. So these things change a little bit with this. And in fact, when let's say that we are interested in precise estimation of Ks, Ka and Ke, these two parameters, rates of absorption and elimination. And when you solve this, now numerically only, that's what we get. Here is the infusion time over some specified time, and then A goes down, but the metabolite is increasing over time and then decreasing. And this is the curve. The red curve here is what is really interesting. And these other curves, this is a very, these dashed are derivative functions for this. So that's how it looks. PK. PD. Well, again, depending on what kind of PD problem we will encounter, there will be different models. We just took as an example here the simplest standard Emacs model. Here I put X, which is dose, but actually what I observed, there may be almost anything here. Emax is the maximum value for a sort of asymptote, and the EC50 is the value of x at which Emax is, you have half of this effect. This is a curve for some values of Emax and EC50. They may have different shape depending on these two values, maybe more sigmoidal. That will vary depending on the parameters. And what we are interested in is actually estimation of these two parameters. Okay, now what we have here. Here we have back our PK model, which is now what I said, what I'm interested in is the concentration of plasma in blood. So concentration on the metabolite in plasma over time for a given dose. And here is the Emax model, but instead of x, I just put this function, the concentration function. So now this depends on time. And altogether, it's changed the shape to this. And these two lines are in our derivatives. So that's my Emax model, or perhaps I should say PK-PD model, but as a function of time. And here I will say now, here my design of experiment comes in. So that's the usual Xi. So I'm interested in times when to measure, and with what proportions of measurements to get the estimates, the precise estimates. Okay, so these are the models. So what are the responses we actually observe? Let's look at it again. First, we observe concentration with some error, and we observe this Emax model, but this is like this depending on time now, with concentration included here. And we also observe this trinomial response, y0, y1, y2. So that's all what is possible, what we can observe in this trial. And then in the case,
sorry, I find your notation confusing.
yes?
I see a subscripted PK, a subscripted PD, and then subscripts 0, 1, 2, and I can't reconcile them.
This is a triangle. This is for
That I understand. So how does it coincide with the PK and the PD?
these
What
are
does it
different
mean?
y's. These are different. These are continuous.
Call him Y3 and Y4.
Those are other things.
Yes. Yes. This is PK.
Oh,
This
they're
is
not Y0, 1, 2.
No.
I see.
that's
Okay.
my expected
No, I
YPK,
understand.
and this is my expected YPD.
Okay.
Now, in the case, yeah.
Sorry, when we ask questions about the model, so assume all the PK parameters are patient specific, right?
Yes.
Not in the notation, but in the intention, I assume.
What do you mean by perhaps patient specific?
Additional
Yeah,
calculation
patient
variabilities.
Every patient has different PK parameters, I assume.
Well, yes, but we just put this error here for that, really. So that's not like-
It's the same for everybody. That can't be the case.
This is different.
I mean, everybody's got
This
their
is
own-
different.
So you don't have an interpatient variability?
No.
There's no patient-to-patient variability.
No.
Oh my God.
Okay. Let me just continue, if you don't mind. What I want to say at the case step, what is the design? Is the dose, which was derived in the previous step as optimum, and this Xi, which was derived in the previous step as an optimum. That's the design at step K. Of course, this K minus 1 will be somewhere here, okay, and Xi will be the T here and here, and also the parameters will be updated. So from step to step, we assume that you have a cohort of size 1. That's what we get. This is adaptive design. Okay?
So step is basically the cohort size.
Yes.
Every time.
Well, no.
Observe
Well,
outcome.
Yes. From you start with cohort 1, observe, update, and so on.
I
Now-
want to ask, sorry, another question. So there's y, you go one slide back. Y0, Y1, and Y2 then nowhere related to YPK, YPD, because usually, you know, efficacy and safety, they are related to them.
The dose is here, but
Measure,
this will depend on dose, and this depends on dose.
and what are their, what do you mean by no efficacy? So do you put some threshold? What do you mean by no?
Well, if there's no efficacy, means that there was no positive response to drug, basically.
At all. And safety, what does it mean for you? No.
Safety, well, this is Y2 here is a probability of toxicity, so we don't want it to be higher than some value. That was
The probability, not the biomarker itself.
no. Okay, so this here you have stars, these stars here. So what are they? Why do we have stars? This because we've chosen at each step, we choose these values in some optimum way. So what are these optimum criteria, optimality criteria? Here we take this simply what we do is just take psi1 and take the dose which maximizes psi1. In Janke, there was a difference between psi1 and psi2 with some parameter gamma between 0 and 1, and we find that this doesn't work any better, and it's just good enough to look at psi1.
What was
So,
psi1?
psi1 is probability of efficacy, and this is probability of toxicity. And this delta 1 is just puts a limit on toxicity probability. So it's an indicator function, and this will be 1 or 0 depending if toxicity is less than some pre-specified value pi 0 or not. And then X star is the argument of the maximization of this. Basically, as I said, we take psi1, but within this region. And this is called, the authors here call it biologically optimum dose. So I call it a biologically optimum dose. You can discuss this. Is it really biologically? I don't know.
You probably defined earlier. So here the efficacy is really efficacy, no toxicity.
That's right, yes.
And it's the paper also had the same definition.
That's right. Now, that's for these responses Y0, Y1, and Y2. For PK estimation, actually PK-PD I should say, we that should be PK stroke PD. We take the optimality, standard thing, maximization of the determinant of the information matrix. Now for these 4 parameters. And here we define efficiency of estimation because we want to have the efficiency high. Slightly different than what Antonio presented today in the morning, because here we have psi star in the numerator and denominator. So what's the difference is, well, I mean, in both cases is the optimum design, but this depends on the dose, and here is the best design over all possible doses. And this is just a given dose, whichever you want to check. So that's what we understand by the efficiency. Okay, so how do we approach this optimization problem? In this way, as you see here. So we maximize over dose the probability of efficacy and no toxicity. Okay. Subject to that there's the probability of toxicity is lower than pi 0, and that efficacy of the optimum design psi, this is the time points given a dose, is not less than some pre-specified value eta.
But psi star again is a function of x, right?
Yes, it depends on x. And here you have the scenario which we look at. So this is the probability of, for some given values of parameters, obviously, of psi1. That's what we are really interested in. This is pi 0. We took a third, maybe a bit high, but that's again maybe changed. Thank you. And what we have here is the determinant, depending on the dose, the determinant of the information matrix for the PK/PD parameters. So you see, for this case, it has a maximum. So first of all, it depends on the dose, and it depends in a non-monotonic way. So the efficiency will also depend in a similar non-monotonic way. And this maximum will pull the choice of doses into this area. And this will pull the doses into these are close, you see that? So that's actually quite, these two criteria together, this maximizing this with having this as a constraint will make, if I want high efficiency of estimation, will make the choice of doses quite limited, in fact. And that's what we see here. With assuming that the eta is in percentage, so the efficiency of estimation is bigger than 60% or 80% or 95%. These are just examples of single simulations. So obviously, you may have completely different output, but we observe that, in fact, there is a common thing in all of them. First, when we...
But this also depends on pi 0.
Yes, of course. Yes, you have pi 0 is this black line here, or here. It changes. It's not pi 0, it's...
It varies, I think it should be fixed.
Sorry?
I thought pi 0 would be fixed.
Pi 0 is fixed, but because the model changes when you update it, the region of the doses changes each time.
What was the pi 0?
Pi 0 was the probability of toxicity. That's fixed, the 1/3. But this is dose here on this axis. So the range of allowed doses will be different from step to step because of the updated model. And that's this black line here, here, and here. Now these pink lines here, and this actually it's down there for 60, and it's down here for 80, and here for 90, and upper limit is here, you see. These two lines come from the constraint on efficiency of estimation. So if you want high efficiency of estimation, your dose range gets very limited. And that's because of the shape, obviously, because of these shapes here. So if you want to have high efficiency, you need to be around here, not nowhere else. And that's what comes out. But that means then in this case, you get very quickly, reasonably quickly, to a dose which is actually that good dose you want. And here it wasn't too, it was quite quick too. And there's more of a little bit of variability here. And then you get into the good dose. This range here, whichever, is a good range, I would say, of doses. This, what is this now? Here you have design points for estimation of PK/PD parameters, and there are 3 points, and they will depend on dose. And these are the time points here for this dose, for this dose, for this dose, and so on, and the weights. So that's clearly the dependence of these parameters. And then if you look at, we've done just a very few simulations, and Maciej was doing it last week in tea break, so we didn't have that much time to do thousands of simulations. So there are very few only. But you can see already a pattern. With this efficiency constraint, you have a larger cloud of points, and with this much smaller cloud of points for K, A and KE, and similar for Emax and EC50.
What is
These are the simulation results, estimates of parameters. And the green and red points are, the red is true, green is estimated, mean. And then the design. So after of these few simulations, you get these designs, the frequencies of the dose levels applied. And again, depending on the constraint. And the constraint is here. So if you want a high efficiency, you will be limited to these doses more than in this case. You have some more doses around here and here, and also some toxic doses. You don't have any toxic doses here. And this picture is the same. It's not the same. It looks similar, but this shows what was recommended and how often as a best dose for further studies. So conclusions. As I said already, this is more relevant where patients are included in the study rather than healthy volunteers. And patients are already treated quite a lot with efficacious doses, which may be important because we've heard today a few times that the prior information from early phase is so much important for starting next phase. And that would give this prior information. Fewer potentially toxic or non-efficacious doses. Some information on the response is already gathered. So that's what I've said already as well. And I think that incorporating this information actually changes the dose selection. And I've showed these pictures, excuse me, let me come back briefly, with 50 cohorts here, but it could well be stopped much earlier. We just done 50 and stopped, but there's no reason just to put the stopping criterions in such a way like if you have, say, 10 patients or cohorts treated with the same dose, stop and take this dose as BOD, and that would be much faster than 50. So that could be done. And the last one, well, if you have a reasonable constraints, you can get reasonable results. That's obvious. Thank you.
Any questions, comments?
So you have a cohort size of 1, right?
No, these were cohort size. In the simulations were size 3.
I see. So is this like a likelihood-based approach?
For estimating alphas and beta, we took some prior distribution, but that was non-informative prior. And for estimating K's and Emacs, we used just the least squares estimation.
Oh, sorry. I didn't understand how you have your efficiency, which depends just on one variable, on X. So what does it mean? Because the efficiency should depend all.
Because it was efficiency. That's yes, that's a different definition than what's the standard definition. Here we have optimum design in numerator and denominator, but this optimum design will have different value, not the design. You will get a different value of the determinant for different doses. So first we take the maximum one in the denominator, and then for any dose, if you want to change, what would be for, say, dose 1.2 or something.
Question is here.
Come to the previous page. Yeah, about point 3. Again, my real life is limited, so I'm not quite sure the real life. But theoretically, if you think about it mathematically, at least you need to put some condition on y0, y1, y2, right? I mean, for example, if all doses all the results y0, the MLE or whatever least square can I estimate?
If all doses were...
If all medicines don't see anything, you don't get any information. There must be some side condition must be imposed.
Yes, what would that be here? Yes, it's not an impossible
It's a
scenario.
non-informative prior, it probably wasn't, what was it exactly?
We
Because
had
it
just
might have regularized it. it's nice.
we had just a uniform prior, but yeah, well, I would have to think about it. We never came across a problem, but indeed,
Yeah, that's
What
when you
would-
side 0, side 1, side 2, somehow. The 100%, the other 2 is 0, you don't see. Somehow related whether it's estimable or not.
yeah, we'll need to look at that. Yeah.
Well, do you have a question?
I want to understand from the very beginning what we measure or what kind of measurements we have from each individual. So it's y1, y or y0. So we have three
Well, in each
three events: success
Good guy, bad guy.
and no success. Then so
It's one trinary event.
yeah, so three discrete variables. And plus to the, do we have measurement of two curves
Yes.
for each individual?
Well, over
Because
time,
we
yes.
talk about PK model
Yes,
and then
yes,
PK model.
Yes,
So,
we measure the
then-
concentration
so we have
And-
three binary variables, which is
and
absorbed
say
in each
you may
individual.
measure pulse or whatever.
Just I repeat for myself. So we have three binary variables
Not
and
two, one categorical variable.
One categorical variable,
And then
yes.
two PK.
Trinary. A
But
trinary.
PKPD, the whole curve.
That I don't
Yeah.
know.
That's what I'm
That's
trying
what-
to say.
I don't know. I don't know if it's 17 measures and
Sorry.
That's a decision, how many you want to measure.
So do I have the whole curve here, or you pick up some kind of
Well,
point?
when you estimate the parameters, you have a curve.
But you
How
only
many
observe it at
measurements
certain
on
points,
each
it's a
subject you are
decision.
doing? Do you want to
Three.
do optimal? Okay, so
Yeah.
you didn't say that. So on each subject, how many times you measure, take the blood for PK?
Well, the optimum number of times, which is 3. With, yeah,
What is optimal for your model?
for the model, yeah.
Because you have just fixed effects here.
Yes.
So does,
And that's structural.
So does the PK, PD model, does it, in your design state, it obviously impacts what time points you choose blood from. Does it impact the dose you choose too?
Yes,
So
because
is that good? Because that's might contradict
It constrains the goals.
it
right?
constrains the dose.
might
So you
be
don't
looking
want
for a
to
dose of
be
different-
with the dose which will have, say, too high effect on your pulse. So.
But you just wanted to be efficient there, right? You wanted a good estimates of the PK parameters that might contradict the goal of getting a good dose from a clinical perspective.
Well, from the clinical perspective, the good dose would be the peak of the psi1. It may, in some cases, it may be that the peak will be outside the range of efficiency of estimation, doses for efficient estimation. Then you'd have to take the borderline one. Yeah.
So I think it could be an interesting design for what you often do is like dose escalation to get to a good dose, and then they're usually writing the protocol, and then we'll have an expansion cohort to learn about PK/PD. So maybe you could do the optimal PK sampling for that expansion cohort. Are you objecting to do that for everyone, right? And that you determine the dose escalation.
I guess I was worried the dose driven by the PK considerations might drive you to a place you don't want to go from the efficacy.
Because you want to see interesting things even if they're not good for the patient.
I see. Well, obviously. But within limits, yeah.
I mean, you had these two curves, didn't you? Which
Yeah.
had maxima in much the same place. So
In
I
this
suppose
case, yes.
Yes, I suppose the discussion is partly what would happen were the two maxima to be more separated.
Well, of course, one that's a constraint. That's a constraint only. Of course, as soon as you see that the constraint is nonsense, you may update it properly.
Please.
Have you tried to simulate 100,000 iterations rather than 50?
I told you that we
The
had
suspicion is
been
that you
doing
can-
it in the tea break
Yeah, but
last
the
week.
suspicion is that you will converge to some degenerate design. And another question, what is your asymptotic design? Is it optimal design for all your parameters?
Degenerate design on the PK or on the doses?
On the doses, because she was showing the doses. And
We don't know what are asymptotic properties. The only thing we observed was that this dose was converging to the best dose.
Yeah, but that's very bad. That's exactly the question.
Why it's bad? I don't see why
Because
it's bad.
Degenerate design means you cannot estimate anything.
No, not in this case.
I think these are good discussions, but maybe that's no, it's wonderful. I think it's interesting. Let's keep that for the Weinen